My name is Michael Trebino. I'm a PhD candidate in Dr. Fitnat Yildiz's lab at the University of California, Santa Cruz in the Department of Microbiology and Environmental Toxicology. In the Yildiz lab, our research focuses on understanding biofilm formation through the model organism Vibrio cholera, the causative agent of the diarrheal disease cholera. Vibrio cholera exists in the aquatic environment both as free-swimming cells and as surface-associated biofilms, which are multicellular communities encased in an extracellular matrix. Humans are infected when they consume contaminated food and water, where Vibrio cholera then colonizes the small intestine. Cholera toxin leads to diarrhea, shedding Vibrio cholera back into the aquatic environment. Now, Vibrio cholera remains a major public health problem around the world where access to clean water is limited, resulting in upwards of an annual 4 million infections and 140,000 deaths. Biofilms enhance Vibrio cholera's environmental survival and infectivity. Therefore, it is important to understand how biofilms are formed and how biofilm formation is regulated. Biofilm formation happens through attachment, matrix production, maturation, and eventually dispersal. Work done by our lab and others determined that a critical regulator of this process is the small molecule cyclic di-GMP, which is produced by diguanylate cyclases and degraded by phosphodiesterase. I studied one of these C-di-GMP signaling systems, the VPV system. This is a three-component signal sensing system with VPVC, a DGC, as the output. Work from our lab has shown that the VPV system is important for biofilm formation in Vibrio cholera, and today I'm going to tell you about my work on the regulation of the VPV system. Since VPVC produces C-di-GMP, I investigated the impact of C-di-GMP on VPV expression. Using a strain that can produce cyclic di-GMP upon induction, I found that increased cyclic di-GMP resulted in an increase in VPV expression. Two major C-di-GMP receptors, VPST and VPSR, are important for biofilm formation in Vibrio cholera. When deleted, this led to a decrease in VPV expression, particularly in the exponential growth phase where C-di-GMP is the highest. Using a transposon mutagenesis screen, I found the alternative sigma factor RPOS also regulated the VPV system. RPOS is crucial for VPV expression during the stationary growth phase, where RPOS levels are highest. Additionally, reintroduction of RPOS rescued VPV expression. To determine if RPOS can impact cyclic di-GMP levels in Vibrio cholera, I quantified cyclic di-GMP using mass spectrometry. I found the loss of RPOS greatly decreased cyclic di-GMP, while reintroduction of RPOS rescued it, indicating RPOS can influence the global cyclic di-GMP pool of Vibrio cholera. Additionally, I found the deletion of RPOS in either the VPST or VPSR mutant led to a further decrease in VPV expression, suggesting RPOS acts through an independent pathway. In this movie, you can see VPV expression in a mature biofilm. In the graph to the right, we can look at VPV expression as a function of biofilm depth. Expression is the highest within the biofilm near the substratum. Deletion of VPST, VPSR, or RPOS led to a decrease in overall biofilm thickness, VPV expression, and the spatial stratification of VPV expression. Based on my work, this is our current understanding of the VPV regulatory network. VPST and VPSR form a C-di-GMP sensing regulatory circuit, and RPOS forms a stationary growth phase regulatory circuit. And these circuits are important during biofilm formation. And with that, I'd like to thank ARC's Dr. Fitnat Yildiz and our lab for making this work possible. Thank you for your time.